The MethylCollector MBD Capture Kit is an enrichment-based assay that selectively captures DNA fragments containing CpG methylation. Starting with either digested or sonicated DNA, it makes use of a methyl binding domain protein, which can selectively bind to methylated CpGs. Magnetic beads are then used to capture the methylation containing protein DNA complexes. The enriched DNA can then be eluted from the beads and the methylated DNA fragments analyzed by techniques like qPCR or NGS sequencing to reveal regions of methylation in the samples. DNA methylation is an epigenetic modification that is an important signal for the normal regulation of transcription and chromatin structure. Alterations of DNA methylation have been shown to be involved in both normal biological processes and in disease. So you could imagine there's tremendous interest in understanding the relationship between these methylation states and how they affect these processes for the possible treatment of disease. Customers have used the MethylCollector assay to analyze the methylation status of candidate genes related to a whole host of conditions. For example, researchers have found increases or decreases in methylation can change the expression of genes that cause certain cancers. They've used it to determine how CpG methylation changes affect the progression of Parkinson's disease. They've also used it to look at how behaviors like smoking, diet, or exercise can affect DNA methylation and aging. Once the regions of interest are identified, further analysis can be done to determine what's causing the methylation changes. They can look for possible treatments, which could reverse methylation changes, or to find biomarkers that can be used to detect disease in the early stages.